Hello students, today we will discuss a new topic, that is microscopy. What is microscopy? Microscopy is the technique used to visualize micro objects. Okay, this is a technique to see micro objects or objects which are very small in size, ठीक है, which cannot be seen through naked eye. So those are those objects which are very small, that cannot be seen through the naked eye. For that objects, microscopy is used to visualize it. Okay, है the technique is used to study cells and subcellular fractions. That means microscopy is used to see a small object like cells and subcellular fractions, that is the particles or molecules present within the cells. ठीक है, to study the cells or the particle and molecules or cell organelles present within the molecules within the cells are can be visualized by using microscopy, and the tool used for microscopy is called microscope. ठीक है, okay. So there are two types of microscopy, optical microscopy and electron microscopy, or there are two types of microscope, optical microscope or electron microscope. Okay, this is the basic classification. What is optical microscope? When the source of illumination of the object is light, that means light is used to illuminate the object, then the microscope is known as optical microscope. And when electron is used for illumination source illumination, then it is known as electron microscopy. ठीक है, by optical microscope we can see the object of the size of about 1 micron. ठीक है ना. That means के an object which is as small as 1 micron can be visualized or can be seen using optical microscope. ठीक है, so that means the capacity of optical microscope is up to 1 micron. That means it can see the object of the size of 1 micron. Okay, but the capacity of electron microscope is much more than the optical microscope. By using electron microscope, one can see the object of the size of up to 1 nanometer. That means as small as 1 nanometer size object can be seen through the electron microscope. Okay, and the basic component of microscope can be divided into two groups. One is the light source and the another is the lens, which is used to magnify or to focus the light on the object. Okay, that is the lenses. Okay, in optical microscope the lenses used are made of glass, and that means glass lenses are used. But in electron microscopy, electromagnetic lenses are used because the source of illumination is electron. Okay, and for resolution limit is 0.2 micron, and for electron microscopy the resolution limit is 1 nanometer. That means the resolution limit is much more for electron microscopy than the optical microscopy. We will discuss the resolution limit. That the resolution limit is the power of optical microscope. ठीक है, that is it can visualize or distinguish two objects present some distance apart. Okay, can visualize the two objects or distinguish two objects present some distance apart. So for optical microscope it is 0.2 micrometer. That means that two objects which is present at a distance of 0.2 micron or placed 0.2 micron apart, that two objects can be distinguished as two objects, okay, or two entities, okay. And if these two objects are present at a distance which is less than 0.2 micron, then these two objects or the entities cannot be distinguished as two different entities by the optical microscope. But in the case of electron microscope, if the two objects are present as close as 1 nanometer, can be distinguished as two different entities or two different points, okay. That means the resolution power is also equivalent to the size of the object or distance between the 2 points which can be visualized, okay, as two different points. For the naked eye, the resolution limit is about 100 micron. That means if the two objects are present 100 micron apart, that two objects can be distinguished as two different objects or two different points. This is the meaning, or this is the resolution explanation of a resolution limit. For the optical microscope, it is 0.2 micrometer. The distance should be 0.2 micrometer, but for this is the 1 nanometer, okay. So this is the difference between optical microscopy and electron microscopy, okay. Now we will go through the diagram of the two type of microscope. This is the light microscope or optical microscope, and this is the transmission electron microscope. In the transmission optical microscope, here is the source of light, maybe a lamp or a laser or an LED, and this is the light passing on the, this is the lens system, okay. First lens is condenser. The function of the condenser is to focus the light on the specimen or object which is placed over a glass slide, okay. This is the glass slide and this is the object placed over the glass slide, okay. And this light when falls on the object and goes on to the objective lens, okay. Objective lens must magnify the size of the object and magnify the size of the object, okay, and then focus it on the eyepiece, and then eyepiece focused it on the eye or digital camera. But in the case of electron microscope, these are the lenses, and that's the source of electron, that is the electron gun, from where the electrons are emitted, okay. And these are the electromagnetic lenses, okay. And this is for electron microscope, the specimen are placed over a metallic grid called EM grid. This is made of either a silver grid or a gold grid, okay. And this is the focused light, and then it is visualized. Unlike the optical microscope, the object cannot be visualized through eye, but rather it is viewed on a screen, on a viewing screen or a digital camera, whereas in optical microscope we can visualize the object through our eyes as well as digital camera, ठीक है ना. One thing is important कि magnification power by optical microscope, the optical microscope has maximum capacity, or it can magnify the object up to 1500 times. whereas the electron microscope, the size of the object can be magnified up to 200,000 times, much more than the optical microscope. Okay, this is the difference between the electron microscope and the optical microscope. Thank you very much.